Hi, and welcome to this video on basic genetic concepts. If you don't have an understanding of cell division and the events of mitosis and meiosis, you probably want to go back and make sure that you have that before you move into this video. Otherwise, some of it may not make sense. The key points in this video are why the events of meiosis and fertilization lead to predictable genetic ratios, and how we can use the understanding of genetics to determine the ratios of traits in offspring. When we talk about basic genetics, what we're really talking about is Mendelian genetics. This is named after the gentleman who first explained these mechanisms of inheritance, a gentleman by the name of Gregor Mendel, who was a monk in the Austro-Hungarian Empire in the 1800s and who did a lot of work with breeding of pea plants in the garden of the abbey in which he served as a monk. Mendel didn't actually know what he was doing when he was doing it, but it turns out that he had a really great system for investigating basic genetic interactions through the use of pea plants. Pea plants are a great model organism for genetics. They're easy to grow. They don't require a lot of space. Every pea is an offspring, so you can get a lot of offspring. It does not take a long time to produce a lot of offspring. It's much easier to see mechanisms of inheritance in a model organism like a pea plant than it would be in something like an elephant, for instance, where the generation time is many decades. The first thing that Mendel did was he developed pure breeding lines of pea plants. A pure breeding line of pea plants is one that has a trait and all of its offspring have the same trait. For instance, if we want to talk about pure breeding white flowered pea plants, this means that the pea plant has a white flower and that all of its offspring, if you grew them and planted them, would all have white flowers as well. Mendel developed 7 different lines of pure breeding pea plants, and you can see them in this chart down at the bottom right of this diagram. So for instance, you could have round seeds or wrinkled seeds, or yellow seeds or green seeds, etc. All of these traits were very obvious and very easy for Mendel to do his experiments with. Let's walk through an example of what Mendel did using flower color. So for instance, Mendel would develop pure breeding purple flowered and white flowered pea plants. And then what he would do was cross those plants. And when he did that, he found out that all of the plants that were produced were purple. This was different than how most people thought about inheritance at the time. Most people at the time thought that inheritance was a result of blending of characteristics. If you took a purple plant and you crossed it with a white flowered plant, you might get like a lighter purple, maybe a puce plant. But that's not what Mendel found. He found instead that all the offspring from a cross like this would be as purple as the purple parent was. And so we should talk a little bit about the terminology that we're going to use here. We're going to call this cross between parents the P generation. And then we're going to call the resulting offspring the F1 generation. Every time Mendel did this for all of his traits, he would always find the same thing. Like if he crossed constricted and full pods, for instance, all the offspring would be constricted, or axial and terminal, all the offspring would be terminal, or yellow seeds and green seeds, all the offspring would be green seeded. And so that was kind of interesting, and already started to show that the way in which traits were inherited was different from how a lot of people thought that they were inherited. But then what Mendel did was he would go one step further. So he would do an F1 cross. So these are F1 purple. These are the offspring of that previous purple plant cross. And when he did this, he found out that he would get purple, but he would also get white. And again, this was always the case. In the F2 generation, resulting from a cross of the F1s of this pure breeded line, you would wind up getting back the parental trait. But what was particularly interesting was the ratio in which you would get these back. You would get them back in a 3/4 to 1/4 ratio. Every time you did this work, you would get the same thing happening each time. And so let's go in and let's see if we can use our modern understanding of genetics in order to understand what was going on in these crosses. What we have here is the purple flowered F1 cross, but now we're looking at both the physical trait and the genes that make it up. So we're going to get some terms in here. We're going to call the physical trait that an organism has the phenotype. And we're going to differentiate that from the genes that the organism has, which we're going to call the genotype. Any sexually reproducing organism is going to have 2 copies of every gene, which we're going to call alleles. One allele comes from the gamete that your biological mother produced, and the other allele comes from the gamete that your biological father produced. During sexual reproduction, those 2 gametes combined during fertilization, which gives rise to your particular genotype. Understanding this, let's go back and take a look at what was happening in our crosses. But in order to do that, we need to keep track of the alleles that lead to these 2 different traits. What we'll do is we'll call big P purple, and little p we'll do as white. Since these are pure breeding plants, We know the purple flowered parent is big P in terms of its genotype. And we know the white flowered parent is little p. And since big P can only make big P gametes, and little p can only make little p gametes, our resulting F1 offspring are going to be big P, little p. And so we're going to need some other terms as well. We're going to say that organisms that have 2 copies of the same allele for any trait are homozygous, whereas organisms that have 1 of each copy of an allele for a trait are heterozygous. The prefix homo means the same, the prefix hetero means different. In Mendel's P generation crosses, he's crossing 2 homozygotes, homozygous purple with homozygous white, and he is determining that all of the offspring are purple. But we know genetically what's happened is that the purple parent has contributed a purple allele, big P, and the white parent has contributed a white allele, little p, leading to the F1s all being heterozygous. This demonstrates the idea of dominance. In genetics, a dominant allele is the one that shows the trait in the heterozygote. Even though these F1s have the allele for both purple and white, the fact that they're showing purple and not white demonstrates that purple is dominant to white. This is going to be common in all Mendelian inheritance. Anytime we're talking about the dominant trait, it's the one that determines the trait of the heterozygote. The other trait, the one that does not show up in the heterozygote, is known as the recessive trait. And so the convention is always going to be to write the dominant allele as a capital letter and the recessive allele as a lowercase letter. The other thing that Mendel's experiment is showing in the F1 generation is the idea of segregation of alleles. In other words, when we make gametes, we only contribute 1 allele to that gamete. The other one is not contributed. The alleles separate from each other, or they segregate from each other. Understanding this, we can get a handle on what's going on in our F1 cross. Since genotypically our F1 plants were both big P, little p, when their alleles segregate to produce gametes, there are 4 possible combinations that they can have. You can wind up having the dominant or big P combined with another dominant P. This would lead to homozygous dominant genotype. You could have a big P combine with a little p. This would lead to a heterozygote. Or you could have 2 little p alleles combine in the homozygous recessive. Because 3 of the 4 possible combinations result in the phenotype of purple flowers, you'll wind up with a 3/4 to 1/4 phenotypic ratio of purple to white. If you wanted to look at it genotypically, our ratio would be slightly different. You'll get a 1/4 to 2/4 to 1/4 ratio of homozygous dominant to heterozygous to homozygous recessives. This is always the case in Mendelian inheritance. Anytime we're looking at a trait with this basic relationship of a dominant allele and a recessive allele, the inheritance of the traits will obey these rules. The other thing that Mendel did was he looked at the combinations of different Mendelian traits and how they're inherited. The prior cross that we looked at with flower color is what we called a monohybrid cross. It only looked at 1 trait. Mendel also investigated dihybrid crosses that looked at 2 traits. This example demonstrates the inheritance of both seed color and seed shape, where yellow seeds are dominant to green seeds, and round seeds are dominant to wrinkly seeds. Mendel's method remains the same. He has a pure breeding yellow round seeded pea plant that he's crossing in the P generation with a pure breeding green wrinkled seeded pea plant. And all of the offspring that are produced in the F1 generation are all yellow and round, as they should be because they're all heterozygous, so they'll show the dominant traits. He then crosses the F1s with themselves, and because there are now 2 traits, there are 4 possible combinations of gametes you can make. You can have gametes that are big Y, big R, little y, big Y, little r, and little y, little r. These 4 combinations of gametes from one parent combined with 4 combinations of gametes from the other parent to lead to 16 possible genotypes, which are all shown here in this diagram, and 4 possible phenotypes in a predictable ratio that you can see down here at 9:3 to 3:1. In order to understand what's happening here, let's revisit our understanding of the events of meiosis, and particularly the segregation of homologous chromosomes during metaphase 1 of meiosis. When chromosomes line up during meiosis, they line up independently of each other. We assume that in this diagram, the red pair is the pair that came from the biological father, and the blue pair is the pair that came from the biological mother, the way in which any one pair of homologous chromosomes lines up has no impact on the way in which any other pair lines up. They are independent events. In other words, the chromosomes assort into their successive gametes independently of each other. This then leads to equal production of every possible combination of unlinked gametes on these different chromosomes, which then leads to the mathematics that are involved in this process and these 16 possible combinations. What's nice about this is that these are independent probabilities. And when combining independent probabilities, the chance that any two independent probabilities will occur simultaneously is equal to the product or the multiplied value of the two independent probabilities themselves. Let's see if we can use this independent probability rule in order to work our way through this particular problem. In pea plants, purple flowers are dominant to white flowers, and yellow seeds are dominant to green seeds. In a cross between two pea plants that are heterozygous for both flower color and seed color, what fraction of the offspring will be white flowered with yellow seed pods? Pause the video and see if you can answer this on your own before we work through it together. All right, well, let's take a look and see what we're doing. We're crossing two pea plants that are heterozygous for both flower color and seed color. So they're both big P, big Y, little y. This is our parental cross, and we want to figure out what our F1 cross will look like. Because these are independent probabilities, let's break them apart. Let's do the flower color cross first. We're going to cross big P, big P, little p. So here's our Punnett square for that cross, and we can just bring down and across our alleles. And then let's look at our seed color cross as well. So we're going to cross big Y, little y. So here's the Punnett square for that one. And we'll do the same thing again. And now we can just find the individual probabilities of these two traits that we're looking for and multiply them by each other. Our white flowered plants are going to be present 1/4 of the time in our flower color cross, whereas yellow seed colored plants are going to be present 3/4 of the time in our seed color cross. So the probability of having offspring that are white flowered with yellow seed pods is going to be equal to 1/4 times 3/4, the independent probabilities of each one, which is 3/16. We would expect that in this cross, we would produce 3/16 of our offspring that would have both white flowers and yellow seeds. That's the power of independent probabilities, and why you do not have to draw a Punnett square with 16 different boxes in order to get the answers to most of the genetic questions that you'll be asked. You can simply find the independent probabilities and multiply them by each other in order to figure it out. As we wrap up this video, let's make sure we can check our learning with the following things. Make sure that you can explain the Mendelian concepts of genotype, phenotype, dominant, recessive, homozygous, and heterozygous. Make sure that you can connect the Mendelian genetic concepts of segregation and independent assortment to events that are occurring in meiosis. And finally, make sure that you can use your understanding of Mendelian genetics to predict and explain the ratios of traits in the offspring of parents with particular genotypes and phenotypes. If you can do those things, you're doing great. If not, that's okay too. Take a moment, pause the video, write down any questions that you have so that you can get the help that you need. Thanks again for watching this video. I really appreciate it. Have a good day.